Thank you very much for the kind invitation. And for me, it's a pleasure and an honor to be together with you this afternoon. And I would like to share with you some of our work regarding the interaction with the microbiome with innate immune responses. And I hope that you can see my screen, hopefully.
Yes, we do.
Oh. Yes. So I would like to discuss with you regarding the role of the microbiome as a factor that influences variation and adaptation in innate immune responses. And what do I mean by that? First of all, immune responses differ between different individuals. Of course, all of us are of different heights. We have different length or color of the eyes or color of the hair and so on. And just as we are different in all other respects, we are also different in our immune responses. Some people are more susceptible for some infections or for some other immune-mediated diseases. Some are less. This is just a very simple example of a study in which, for example, people with Candida in their bloodstream are treated with caspofungin. And although most of the people will eliminate the microorganism from the bloodstream under the treatment with caspofungin, 20% of them will still have Candida in their bloodstream by day 4 and 11 by day 7, and 6% even by day 10. So apparently there is a very big difference in which people can eliminate Candida when they are properly treated, in which they undergo the severity of an infection or not. So the question is, what are the factors influencing these immune responses in the individuals, either healthy individuals or individuals with a certain disease? And we started some 12 or 13 years ago, the Human Functional Genomics Project, in which we try to understand disease susceptibility from the immune point of view. So we are interested to understand which are the factors which influence variability in the immune response, and that in turn influences disease susceptibility. And we have started doing this by looking in large cohort of individuals in depth of their immune responses and identifying the factors that influence them. This is, for example, what is happening when a microorganism, a pathogen invades our body. For example, it invades the tissue. Macrophages in the tissue are stimulated at that moment, either by pathogens themselves or by the tissue damage determined by these pathogens, which eliminate danger-associated molecular patterns from the destroyed cells. And these macrophages, which sense the pathogen or the destruction induced by the pathogen, are activated and induce production of either chemokines, which recruit more cells to the site of the infection, or pro-inflammatory cytokines, which at the same time stimulate these cells. These pro-inflammatory cytokines are also released in the circulation. They go to the liver, where they stimulate production of acute phase proteins. They go to the hypothalamus, where they induce processes like fever, somnolence, anorexia, pain, and so on. At the same time, the other cells which are recruited, such as neutrophils or monocytes from the circulation, get activated. They start to phagocytose the pathogens and kill them. And most of the time, this is enough for elimination of the infection. However, in some situation in which after a couple of hours or days, we do not eliminate the pathogen, then T cells and B cells are activated. And they produce, for example, the Th1 cells, interferon gamma, the Th17 produce interleukin 17 to activate neutrophils, interleukin 22 to activate epithelial cells to produce defensins, and so on. And in these cohorts of individuals that we have done, every component that is in a red circle was also studied in depth, and we assessed the function which influence these various components of the host defense. Which are those factors? So we started actually with a number of cohorts of healthy individuals in Nijmegen, in our town where we coordinated the study. We have done 2 cohorts of 500 and 200 healthy volunteers respectively, in which we assess cytokine production capacity and as a mirror of innate immune responses. And then we assessed various factors. We assessed demographic factors through questionnaires. We did transcriptome in the innate immune cells. We did immunophenotyping. We assessed the genetics by doing SNP arrays and so on. But we also assessed the microbiome. And then we asked ourselves, which are the factors which influence the innate immune responses? And first of all, I will start with some very simple things, very simple factors, which are nevertheless very important. For example, age and sex. Age, we know that we are more susceptible to infections when we become elderly. And the question is, what component of the innate immune response is defective in individuals of older age? And it turns out that not every type of function of any type of immune cell is defective. For example, production of pro-inflammatory cytokines from myeloid cells, such as IL-1 beta, TNF, IL-6, is perfectly normal in elderly individuals. However, production of cytokines which are produced by T cells, such as interferon gamma and interleukin-22, are severely decreased in people of older age. So they are defective, and they probably explain part of the defects at the level of the mucosa and why the elderly people get so many infections. On the other hand, for example, the sex influenced also, especially the monocyte production of pro-inflammatory cytokines, and the men produce more. And this can be disadvantageous because, for example, this can account for the increased metabolic dysregulation and atherosclerosis, for example, which is mediated by inflammatory processes in men compared with women. We also assessed which are the genetic factors which are important for the modulation of cytokine production capacity, and we identified 17 different loci in our genome which are very strongly influencing the cytokine production. The locus which influences the strongest the cytokine production capacity in European population is the locus which contains the very well-known pattern recognition receptors, toll-like receptor 1, 6, and 10. And you can see here the p-value. This each point represents one polymorphism, and the p-value for each of these polymorphisms can be read. On the y-axis, you can see here p-values of 10 to minus 25, basically, in the capacity of this polymorphism to influence cytokine production capacity. So very high. Very interestingly, just like a parenthesis, this locus is very important for influencing cytokine production in Europeans, but not in Africans or Asians. And the question was also why, and there was research done, what precisely is going on in Europeans at that locus. And it seems that the variation in this locus is inherited in European populations from the Neanderthals. And the Neanderthals influenced strongly our genetic makeup. I mean, strongly. 1.5% of the European DNA is Neanderthal, whereas, for example, in people with an African ancestry, 0, because there was no mix between Homo sapiens and the Neanderthals in Africa, only in Europe. So these are the 17 independent loci that influence cytokine production. This is a circular Manhattan plot, and you can see here each locus. This one is the TLR1 1610 on chromosome 4, but there were 17 independent loci which influenced strongly cytokine production capacity. So genetics has a very important role in determining our response to pathogens. How about the microbiome now? And we performed in the 500 functional genomics cohort an extending assessment of the microbiome by performing full sequencing metagenomics. And we associated the microorganisms that we identified with the cytokine production capacity. And again, the 6 cytokines that I mentioned before, pro-inflammatory cytokines released by the myeloid cells, TNF, IL-1 beta, and IL-6, and T-cell dependent cytokines such as interleukin-17, 22, and interferon gamma. And when we performed these associations at the species level, we observed effects of several species that influenced either interferon gamma, IL-1, IL-6, and so on and so forth, as you can see here. And these are some examples. For example, one of the strongest association was with Coprococcus comus and production of IL-1 beta, IL-6, and IL-22. as you can observe here, a positive association between Coprococcus comus with production of IL-1 beta and IL-6 by myeloid cells, and an inverse relationship with the production of interleukin-22 stimulated by Staphylococcus aureus. And at that level, we can identify which species are important for the cytokine production. However, at that moment, we thought, well, there are some effects here, as you can see, but they are not that spectacular, honestly speaking. There are some difference. But then we thought, okay, these are the species, but is the species which is the most important in the microbiome, or in fact, is the function of that microorganism which is important? So what do I want to say from that? Let's say that somebody has a production of butyric acid, for example, in the gut. So butyrate, we know it has immunomodulating capacity, but maybe in one individual is one species which is producing butyrate, and in another individual is a totally different species. They have different names, but they have the same function. So maybe just by assessing the name of the pathogen is not enough to understand properly the entire impact of the microbiome on immune function. So this study was done in collaboration with our colleague from the Broad Institute at MIT Harvard in Boston, our colleague from the group of Ramnik Xavier. And in the two panels on the left side, you can see here the variation of the microbiome in terms of species and phyla, and down variation in the metabolic pathways across the samples. Now, what you can observe is the following. On the top panel, there is a huge variation between individuals in the way that they have Bacteroidetes, actinobacteria, firmicutes, and so on. Huge difference. You can see, for example, in the Bacteroidetes, there are people who have 0, and there are people who have 50% of their microbiome from this genus. The same is, for example, with actinobacteria. You have people who have 50%, and you have people who have almost nothing. So there is a huge variation in the names of the bacteria. Now, if we look at the function that this bacteria has, and you can do this actually by assessing the genetics of these bacteria, if you look at the function, and this is the second panel, the panel left under, you can see here, and these are all different functions: glycolysis, mannose, biosynthesis, sucrose degradation, and so on. Now you see that the entire population, despite the high variability in the names of the microbe, it is much more homogeneous. The function of those microbes have to be the same, because otherwise we go into a pathologic state. And you can see now that in terms of the microbiological function of our microbiome, there is much more homogeneity between individuals. Now, we assessed thereafter what is the importance of this microbiome function for the cytokine production capacity, and how much of the variation in cytokine production capacity can be explained by the function of the microbiome. And the number we came with was approximately 5%. You can see here, there are also cytokine productions that are less influenced by the microbiome, maybe only 1 or 2%, and some of them which are around 10%. For example, production of TNF by Candida albicans is influenced, the variability in that production is explained by the microbiome for 10%, but the vast majority are around 5%. So we can conclude more or less that 5% of our variability in cytokine production capacity is dependent on our microbiome. Now, thereafter, we have looked exactly which type of cytokines, which type of biological processes are influenced. And what we have observed very clearly is that it is especially interferon gamma and TNF, which is strongly influenced by the function of the microbiome, and much less IL-1, IL-6, IL-17, which was surprising for us. I would have expected myself that the strongest cytokines influenced by the microbiome would be IL-17 and IL-22, but it turns out not to be the case. It's actually interferon gamma and TNF, which are the strongest associated with a microbiome function in our intestine. These are thereafter some example that this is indeed the case. You can see here association of interferon gamma production or TNF production with abundance of certain microorganisms and certain metabolites of the microorganisms. And then we, for example, tryptophan metabolism, palmitoleic acid, cisvoxanic acid are associated, and we have observed basically that that we can reproduce these effects of these metabolites released by the microorganisms in vitro in experimental studies. Now, this is very, very nice. And then we thought, okay, let's put everything together now. And how much of the variability of the innate immune responses are explained by various components that we have in our body. So, for example, genetics, but also immunoglobulins, hormones, metabolites, microbiome pathways, and so on. And we put everything together and we analyze that. And what we have observed is the following. Genetics is by far the most important single factor. Approximately 25–30% of our variability in the innate immune responses are genetically determined. Now, there are also non-genetic factors which account for approximately 70%, but there are many factors. So it's not one factor, it's many factors which contribute to these 70%. And the microbiome function, as I mentioned, it's here in the middle. It's approximately 5 to maximum of 10%. So this is just as important, as you can see here, as the metabolites in the blood. Another important factor, more important than we expected, was the seasonality. Season was very important. Whereas, for example, sex and age were less important than these factors, surprisingly enough. But anyway, we learned a lot from this. Now, does the microbiome indeed influence the human metabolome? Our metabolites in the circulation, how strong is the impact of the microbiome? Compare with the genetics and compare with the diet, which are two important factors as well. And we did this analysis together with our collaborators from University of Groningen, and we assessed these three factors. And you can observe here, probably not surprisingly, by far, the most important factor for our metabolites in the circulation is the diet. Because, of course, the diet gets us metabolites, provides us with metabolites, so it influences what metabolites are in the blood. So no surprise there. But thereafter, you can observe that basically the microbiome explains approximately 12.8% of the variability in the human metabolome, whereas genetics only 3.3%. So basically, in contrast with what is happening, for example, with cytokine production capacity, the metabolites are especially influenced by diet and the microbiome. Microbiome approximately 13%, diet much higher. And this is the impact of dietary habits on circulating metabolites. You can see here, drinking coffee or alcohol or cereals, red wine, and so on. But as you can see here, very interestingly, alcohol and coffee influence the highest numbers of metabolites. They influence them positively, so they stimulate them. Whereas with other dietary components, such as cereals or dairies or milk and so on, they induce inhibition of these metabolites. So they have very different effects. Okay, how important is this impact of the microbiome on metabolites and then on cytokine production capacity? And to do that, we studied a population in which big changes in diet takes place. And this is a population in Africa, in Tanzania, in which people are currently undergoing a massive transition from a traditional diet in a rural area towards the city, when they eat a Western type diet of processed food. And we have performed a detailed study in population from the city of Moshi, which is an urban area of approximately 200,000 individuals. And we collected there approximately 250 individuals. And then we collected also 70 individuals from the rural area around Mount Kilimanjaro, which is very close to Moshi. And they have a very traditional type of diet and lifestyle. First of all, we looked at the BMI of these individuals. You can observe here, unfortunately, when you look at the BMI, is that the urban people already gain BMI. They become fatter, unfortunately, because of the processed food, because of the number of calories they ingest, and so on. And thereafter, we assessed their immunological profile. First, we looked with RNA sequencing whether they have a different transcriptional profile in their immune cells in the circulation. And we observed very clearly that, for example, there are changes when we compare people between from rural to urban areas. Interferon responses, for example, are much strongly upregulated in urban areas, whereas other components such as coagulation, IL-2 STAT5 signaling, complement, TGF-beta signaling, and so on are stronger present in the rural areas. Why, for example, interferons in urban? Probably because of the population density. The exchange of pathogens and viruses are much higher in an urban area, and this leads basically to a chronic stimulation of interferon pathways. Then we looked at cytokine production capacity in these two populations, and what we observed very clearly is that people in the urban area, so in the cities, produce much more pro-inflammatory cytokines compared with the people living in the rural areas. So basically, they have a much stronger pro-inflammatory profile, which is it's a pity because it means, and that can explain actually our metabolic dysregulation, atherosclerosis, all the diseases of civilization, let's say. We compared also the metabolites between the rural and urban areas, and we observed clear differences between them. Some of them more abundant in urban areas, some of them more abundant in rural areas. You can see here, for example, bile acid biosynthesis metabolites are more in an urban areas. Amino acid metabolism, for example, TCA cycle and so on, more in the rural area. And then we associated these metabolites again with the immune function. And the picture was really striking. The metabolites that were more present in the rural areas were associated with lower cytokine production capacity, whereas metabolites which were high in the people living in urban areas were strongly associated with a pro-inflammatory profile. So indeed, it seems actually that there is an interaction between the diet, the metabolites, and the pro-inflammatory profile, which is higher in urban areas. We also confirmed that through validation experiments in vitro. And then we looked at the impact of the microbiome on these metabolites in our population in Tanzania. We compared again the people in rural and urban area. We compared them also with a microbiome from European individuals from the Netherlands. In the panel C, you can observe a principal component analysis. You can see how different Europeans are from people living in Africa from the point of view of the microbiome, of course, the diet, the genetic background, everything. And then we assess whether there are certain microbiome species which are higher in the rural area compared with urban, and indeed that is the case. There is much more richness actually of the microbiome. You can see here in orange that is much more orange than blue. There is much more richness actually in rural area. And in the end, that was associated also, it was especially phenols and phenol derivatives, which were associated with a calming anti-inflammatory effect on cytokine production, as you can see here in this graph with a negative association versus positive associations of the microbiome and cytokine production. So what can we conclude from that? That in countries undergoing currently rural to urban transitions, which is most of the developing world at this moment. And of course, we have undergone this type of transition also 50 or 100 years ago in Europe as well. There is an association with changes in the diet, an increased BMI, and changes in intestinal microbiome. Unfortunately, this leads to a profile of metabolites in the blood, which is more pro-inflammatory, and that leads to increased pro-inflammatory cytokine profile, which is probably also the substrate of the diseases of civilizations that we face in developed countries. And this will lead actually to new challenges for healthcare, also in developing nations now who are undergoing this transition and who will be confronted actually with an epidemic of non-communicable diseases as well. Finally, of course, I would like to thank you, but also the colleagues of us who did all this work, the colleagues who helped us from Athens, from the group of Professor Giamarellos, but also the colleagues from Hanover, from Groningen, with whom we did a lot of the systems biology studies, colleagues from Harvard, with whom we did the microbiome metagenomic study, and the colleagues from Kilimanjaro Research Center in Moshi, with whom we did our study in Tanzania. Thank you very much.
Well, thank you so much for this elegant presentation. And I mean, all this is really food for thought. So I would like to take the advantage of being the chairman and ask a few questions. So from the presentation, the first striking, what really struck my attention was that the modulation of the microbiome is associated with the cytokine production of Th1 responses, TNF and interferon gamma.
Yes, correct.
Can this explain why Crohn's disease is a Th1 disease?
It's an excellent question. It's an excellent question. It might very well be one of the factors that influences it. We have not done ourselves studies in Crohn's disease, but many people have done it, and they have reported associations of the microbiome with the severity of the disease. Unfortunately, they have not linked it with the immune responses, but I think this hypothesis is very tempting and likely to be true. Yes.
If I may continue, it is obvious that if you have different patients, if I may open a parenthesis, the whole model is based on what we call healthy. Of course, we don't know what is healthy means, but let's assume that the same differences apply for patients as well.
Mm-hmm.
Can this be the case? Can this explain why a response of patients with whatever kind of chronic inflammatory disorder to biologicals cannot be predicted? It can also explain the phenomenon of refractoriness to treatment.
Yes, I think that it can have a role. I think that in cases of the diseases of the intestine, for example, the Crohn's disease or ulcerative colitis or celiac, this impact will be higher than in other types of inflammatory diseases. If we look at the systemic responses, we observe that the microbiome can influence approximately 5%, and in some cases a maximum of 10%. So a relatively smaller effect than other factors. So I think that the microbiome impact on innate immune responses is very strong locally and influences inflammatory bowel diseases. And it will be one of the factors influencing systemic inflammatory diseases, other types of immune-mediated diseases. But that impact of approximately 5 to 10% will be probably smaller than other variations, such as genetics, which will influence 30% of the responses, which is also very variable. Genetics of an individual is also very variable, but also infections that somebody encounters or dietary factors and so on. So I think it's a combination. And the microbiome will be more important, I think, locally, and being one of the important factors systemically, but not the only one.
I have one more question, and I believe that what I'm about to say probably reflects much of the thoughts of people who are in the audience, the majority of which are gastroenterologists. So you know that one of the actually, you know that I'm a fan of probiotics. We have discussed that also several times. So can it be the case that if you give probiotics, although originally the prescriber considers that through this approach, he modulates the microbiome towards a beneficiary response, this could be vice versa?
So,
and also, can this also explain why there is such a heterogeneity of clinical studies with probiotics, where some supplements, they have one probiotic, other have two, other have 10, which means that the more antigens you give, the more harm or benefit you introduce?
Yeah, I think in terms of probiotics is also you're right. It's also a large variability, I think, in the way that we react to probiotics, also in the way that we react to in the way that the probiotics can induce colonization with the strains which are contained in the probiotics. So I think that depending on the microbiome of the host, the probiotics will have a different effects, because for some individuals, their microbiome will be more permissive to the strains in the probiotic, and for other people, those strains will have absolutely no chance to stay, let's say, and have an impact, and they will be eliminated very quickly. So the interaction between the strains in the probiotics and the intestinal microbiome of the various individuals is also very important, I think.
Well, I apologize for getting all the questions or comments from the floor. Professor Rama, please.
Thank you very much for your amazing talk, and congratulations. Just to add a question, I'm pediatric gastroenterologist, and quite often for every symptom, even for gastroesophageal reflux, antibiotics are given very, very often. And my question is, since diversity is very important, could you drive change for long life, this diversity, by giving one or two species of the probiotics, and making not a best effect, but a side effect? Thank you.
Yes, I think indeed the richness and the variability of the human microbiome, the diversity of the human microbiome, is very important for maintaining actually the metabolic function of the microbiome, because I think one of the most important function of our microbiome is to provide us with the right type of metabolites which maintain the health, the recovery of the epithelial cells, the immune tolerance, and so on. Antibiotics have the potential actually to wipe out an important component of the microbiome and disturb the microbiome ecosystem. So from that point of view, indeed, I would use antibiotic when it's needed, but not for everything, obviously. And probiotics can help in improving the diversity, but they are not by themselves the entire answer to the question. Hopefully, when the antibiotic is stopped, diversity returns. Many times it does. If the disturbance was very severe, sometimes it doesn't. So probiotics certainly can have a role, but they cannot supply the entire diversity that is needed, I think.
Other questions or comments? If not, we would like to thank you really very much. That was a great lecture. It was a highlight for our conference. We thank you very much. And as we discussed, you stay committed that in the next conferences, you will continue to support us by your presence and enthusiasm. Thank you so much for being with us this afternoon.
Thank you very much. Thank you.
Κλείνοντας την παρουσίαση, θα ήθελα να πω ότι εκτός από όσους είμαστε στην αίθουσα, είχαμε συνδεδεμένους 58 συναδέλφους διαδικτυακά, το οποίο έτσι με κάνει πάρα πολύ αισιόδοξο. Εγώ σας ευχαριστώ πάρα πολύ και νομίζω ότι είναι ευχάριστο ότι έχουμε και λίγο χρόνο. Άρα...